one of the primary causes of hemolytic anemias are oxidative types of red cell injury. So normally red cells have various mechanisms to deal with oxidative injury, and when overwhelmed we see evidence of oxidant-induced red cell injury, which can potentially lead to anemia if severe enough. And so there are certain endogenous and exogenous substances that animals can ingest. For the exogenous types, and endogenous might be underlying diseases that can result in the production of reactive oxygen species. These then alter some of the hemoglobin to denature it, and it actually has to do with the sulfhydryl groups of hemoglobin, and I'm not going to go into a lot of that. And so hemoglobin denatures, and this causes instability to the red blood cell, and this instability can result in membrane abnormalities, or can actually result in destruction of the red cell within the vessel, or can be phagocytized. So that means that oxidative injury can cause intravascular and extravascular hemolysis. So we can kind of see a mixture. So identifying oxidative injury it's going to require evaluation of a blood serum. No hematology analyzer is going to tell you that you have oxidative injury. And so we see Heinz bodies as one form of oxidative injury, and the other are eccentrocytes. And again, these are both due to oxidative red cell injury. And so Heinz bodies are these little red noses. That is a terrible nose. Let's try that again. Terrible. Well, that's not too bad. But it's a little red nose on the surface of a red blood cell. And again, it's denatured hemoglobin. Whereas an eccentrocyte is, so the hemoglobin, so normally you can see there's our central pallor, at least in a dog. Whereas here, all the hemoglobin sort of fuse together on this one. And then you have this little membrane that's left. It looks almost like a little handbag. So Heinz bodies are actually identified in a really specific way. And I'm going to push pause and I'm going to draw a little picture so you don't have to wait. Okay, so this is my version of methylene blue staining. And so methylene blue staining, which I mentioned it when we were talking about reticulocytes. You take 1 drop of blood, so it's a 1-to-1 ratio of blood and methylene blue, and you let it incubate for 15 minutes or more, and then you make a blood smear. And why you do that is you're looking for either reticulocytes, and so that's what those are, or you can find Heinz bodies. And Heinz bodies on retic stain or on the methylene blue stain are blue. So that is a blue Heinz body. And they show up so much better on methylene blue. But you can also see them on regular right Giemsa or Diff-Quick. So you can identify them, but sometimes you'll get to see that on methylene blue. So the eccentrocyte doesn't really highlight. And the problem with eccentrocytes is they actually look, not that you necessarily know what spherocytes look like yet, but when we talk about spherocytes are small, dark, round red cells. And so sometimes the eccentrocyte, the handle breaks off. So if that handle right there breaks off, it's actually going to look like a spherocyte. But that's not really a spherocyte because a lot of times the membrane will be a little bit irregular. So membrane, that is not going to work. But the membrane might be a little bit irregular, and we actually call these cells pycnocytes. And they look just like spherocytes. And it can be kind of hard to tell, but you don't want to misdiagnose an animal with oxidative injury with immune-mediated anemia, and we'll talk about that more later. So we look for Heinz bodies and eccentrocytes either on our blood smear report or actually looking on the blood smear when we are looking at it ourselves. Other things you can see met hemoglobinemia. That's not something we really talk about on blood work, but this would make your plasma kind of a chocolate brown color, your blood chocolate brown and your plasma very discolored. And you can, obviously, if it's intravascular or extravascular, both of these are going to cause increases in bilirubin and bilirubinuria. And of course, intravascular is also going to cause hemoglobinemia, so red plasma, and then hemoglobinuria, so red urine. And I had mentioned that you can see false increases in MCHC, and of course one of the reasons is because of hemoglobinemia. And another is actually because of the Heinz body, because it's abnormally detected by the CBC analyzer, the hematology analyzer, and so this can be due to the hemoglobinemia or it can be due to the Heinz bodies themselves. So causes of oxidative injury are a little bit species specific. I'm actually not going to number them. Dogs and cats can both have onions and garlic. It's dogs that I mostly think of with zinc, and that's usually pennies minted after 1982. And cats, I also think of acetaminophen, also known as Tylenol. Horses, I've only seen one case recently. They'll eat dried red maple leaves. And then sheep and goats, I've seen rare cases where they can actually ingest copper. So if there's a feed with a lot of copper. So those are all potential causes, and you'll look for the sort of aforementioned things to go along with it to support it.